Group 1. Genetically modified organisms in plants. Just what exactly do these two plants have in common? They have both been altered into genetically modified organisms, or GMOs. But what does this mean? These vegetable genes have been altered to create uniform, cookie-cutter crops that are pesticide-resistant, perfectly colored, large in size, and have a long shelf life. Back in 1992, the first genetically modified tomato was created. This ultimately resulted in a domino effect of genetically modified foods. You're probably wondering exactly how they modify these crops. And if not, well, that's too bad, because we're going to tell you anyway. First, the desired characteristic of a crop is isolated and removed. This is done through a genetic mapping. A gene map is created outlining the DNA sequence and assigning DNA fragments to chromosomes. The scientists then make many copies of these desired gene characteristics. This is done through PCR, polymerase chain reaction. PCR is used to amplify a piece of DNA to create thousands of copies of the DNA sequence. These isolated genes are then placed into the DNA of a new plant's DNA. This can be in one of three ways. The first is a gene cannon, which uses small golden beads as cannonballs. The desired gene is glued onto the surface of the ball, and thereafter the ball is shot into the cell nucleus. The second one is a protoplast. A protoplast is a plant, bacterial, or fungal cell that had its cell wall fully removed using mechanical or enzymatic means. And the third is a soil bacteria. A soil bacteria are bacteria that live in soil to perform important services related to water dynamics, nutrient cycling, and disease suppression. Once the gene is inserted, voila! We have a genetically modified organism. The scientist then takes the plant to make sure that the characteristic has successfully been transferred and that the new seedings also have the characteristic. The end.